Hello everyone, my name is Chioma. Today I'll be talking on a topic I call visualizing the unseen. It's about a problem every biology instructor encounters, which is how do you teach something a student can never actually see? So things like cellular respiration, cell division, molecular interactions, all of them, they all happen at a microscopic scale that students cannot observe directly. And for decades, our main teaching tools have been static diagrams and verbal explanations. So I will be walking you through a conceptual framework for using virtual reality to address this, and it's grounded specifically in cognitive load theory. I'm going to be upfront and say that this is a literature-based synthesis, not an empirical study of my own. By the end of this talk, you will have a concrete set of design principles that you can apply and a clear picture of what the existing evidence does and does not support. So for a quick roadmap, we'll start with the problem itself, then we're going to ground the talk in cognitive load theory. From there, I would walk through what the VR education literature actually shows, which is more mixed than what most virtual reality advocacy suggests. Then I will present the framework itself, 3 design principles, and show how they apply to 2 specific biology topics, in this case, cellular respiration and cell division. I will then close with practical constraints and where this needs to go next. Here's the core problem. Introductory biology asks students to understand things that are quite literally invisible. You can't watch cellular respiration happen with your own eyes. You can't watch mitosis unfold in real time in a lecture hall. So for decades, the tools available have been static diagrams, animations on a screen, and the instructor basically talking through what's happening. That places the entire burden of building a 3-dimensional moving mental model onto the student's imagination, working only from a flat image. And this is not just an inconvenience, it has real consequences. Introductory biology courses, sometimes called gateway courses, they see high rates of disengagement and attrition, and difficulty forming accurate mental models of abstract content is one contributing factor. So the theoretical anchor for this whole framework is cognitive load theory. This was first articulated by Sweller in 1988. The core idea is simple. Working memory has a limited capacity. Sweller distinguished between intrinsic load, which comes from the material itself and can't be reduced without changing what you're teaching, and on the other hand, extraneous load that comes from how the material is presented, and that's the part that instructional design can actually influence. Meyer extended this to multimedia specifically, proposing that we process visual and verbal information through separate channels, each with its own limited capacity. On the right, you can see the basic flow, which is the visual and the verbal inputs, where they come in and they compete for the same limited working memory space before anything gets built into the long-term schema. The design goal for virtual reality content is to represent complex, invincible biology visually and spatially without letting the VR environment itself, its controls, its navigations become a new source of extraneous load competing with the actual content. So, what does the broader virtual reality in education literature, what does it show at the moment? Several major systematic reviews, they converge on a similar picture. The first one is the Natalie et al. 2020, where they reviewed a decade of research across K-12 and higher education, and they found that virtual reality reliably boost engagement and motivation, but learning gains specifically are much more mixed and depend heavily on the design. Luo et al. 2021, after reviewing 149 studies that were spanned over 20 years, they found an overall medium effect but with substantial moderating factors, meaning the effect size really depends on how the VR was built and used. Pellas et al. 2021, they found that visual design and interaction choices, not the degree of immersion itself, were what separated strong outcomes from weak ones. And finally, Radianti et al. 2020 made an important critique. He pointed out that a lot of published VR applications are built around what the technology makes possible, rather than around an explicit learning theory. That gap connecting VR design decisions back to a real theory is exactly what this framework is trying to address. So, this is the part of the literature that virtual reality advocacy tends to skip, and I think that this is the most important slide in this talk. Parong and Meyer, 2018, they had college students learn a biology lesson on how the human body works, either through immersive VR or a desktop slideshow. The VR group, they did worse on the post-test, despite reporting higher interest and motivation. And researchers, they linked that gap to a higher self-reported extraneous cognitive load in the VR group. On the other side, Makransky et al. 2019 found something similar with a science lab simulation. They added that immersion increased presence, but it produced lower learning outcomes than a less immersive version. So the takeaway here is not that VR does not work. It's that immersion and cognitive load reduction are not the same, and the framework that assumes maximizing immersion will automatically help learning is building on a false premise. So if immersion alone doesn't help, what does? This is where the picture gets more useful. In a second experiment, Parong and Meyer had students pause and write a short summary after each segment of the VR lesson, instead of experiencing it as one continuous sequence, and that closed the performance gap with the slideshow group. Then Kligenberg et al. 2020, they found something directly relevant. They studied undergraduate biochemistry students learning the electron transport chain, part of cellular respiration, and found that having students teach the material to a peer afterward significantly improved transfer and retention, specifically in the VR group, not in the desktop group. And then a later study by same team, they found the same benefit from segmenting the content into paced chunks. The pattern across all three studies is consistent. The problem isn't VR itself. It's presenting it as one continuous unscaffolded experience. So one more theoretical piece before we get to the framework itself. Johnson Glenberg in 2018, he worked on embodied condition, and he added something that cognitive load theory alone doesn't fully capture. The idea that learning is strengthened when a learner's physical gesture is congruent with the concepts being taught. Reaching out to rotate a molecule with your hand is congruent with the content. Pressing an abstract button on a controller to do the same thing is not, even though both count as interactivity in a genetic sense. This matters because interactivity in VR is often treated as an automatic benefit. More interaction equals better, but interactivity that isn't tied to the learning objective can itself become a source of extraneous load, especially if students spend mental effort figuring out what they are allowed to do rather than focusing on the biology. This directly shapes one of the three design principles I'm about to walk you through. Now, bringing all this together, here's the framework itself. Three design principles for managing cognitive load in VR biology instruction. First, we look at guided navigation movements through the environment. It follows the logical sequence of the process. So students aren't spending mental effort figuring out where to go on top of what to learn. Then next is scaffolded complexity. That is, stages are introduced progressively rather than all at once. This is the principle with the strongest direct evidence behind it. It's drawing straight from segmentation studies we just covered. And finally is controlled interactivity. Actions are limited to those that directly support understanding of that specific stage, and it draws on Johnson Glebeck's point about gesture congruence, that's rotating a mitochondrion to see respiration happening inside, rather than open-ended manipulation of everything in that stage. So let's make this concrete with our first illustrative example. Let's use cellular respiration. So applying the framework, a viral molecule or a viral module would be structured around the sequence of glycolysis, the citric acid cycle, and the electron transport chain, with students moving through each stage in order, rather than being dropped into an open environment containing all three at once. So at each stage, interactivity would be limited to actions that support understanding of that specific stage, like observing electron movement along the transport chain, rather than free exploration of the whole cell. And importantly, this isn't purely hypothetical. Barrow et al. In 2024 ran a pilot VR study on exactly the citric acid cycle, structured as 8 sequential activity levels with undergraduates. And also, Kleinberg et al. In 2020, they studied the electron transport chain, specifically with undergraduate biochemistry students. And these two existing studies, they structure their content around discrete steps rather than the full pathway at once, which lines up with what this framework proposes. So, second example is cell division. The same design logic applies to the 5 stages of mitosis, prophase, metaphase, anaphase, telophase, and cytokinesis, with students observing and interacting with each stage in sequence rather than all at once. There's existing work here too by Nasharuddin et al., where they developed in-cell VR. That's a mobile-based VR application for teaching human cell division, and they found knowledge gains from pre-test to post-test. So it's worth noting that this was mobile VR, not a headset-based immersive experience, but it illustrates the same underlying principle this framework is built on. So I want to make this framework tangible. So this slide is a mock-up of what an immersive experience page for the citric acid cycle of cellular respiration might actually look like. Applying the 3 principles directly, notice the top bar, stage 2 of 3. So the student already knows where they are in a guided sequence. That's guided navigation. The scene shows one structure, the mitochondria, with one stage of one process, not the entire cell with everything happening at once. That's scaffolded complexity. And the callouts and the rotate prompt are tied directly to what biological happening at this stage. That is acetyl-CoA entering, NADH and FADH2 being released, CO2 as a byproduct, not an open-ended set of things to click on. And that's controlled interactivity. I want to be clear that this is an illustrative mock-up I built for this talk, not a screenshot of an existing product. Before I close it, what's naming the partial constraints that exist outside of instructional design theory? Cost remains real because headset hardware, even at today's reduced price, is a cost that many departments and institutions cannot afford. Then there's also access, where students without personal VR equipment depend entirely on what the institution can provide, which raises real equity issues. And also, instructor readiness matters because faculty who didn't train with this technology, they need meaningful support and time to get comfortable before operating the hardware and designing instruction around it. So where does this need to go from here? First and most importantly, I want to be honest that this is a conceptual synthesis, not an empirical study. So everything I've shown should be treated as a framework to be tested, not an established finding. The most useful next step would be classroom-based studies that directly compare VR instruction built on these principles against traditional visualization using actual cognitive load measures, rather than relying on self-reported engagement alone, since engagement and learning doesn't always move together. Specifically, it would be valuable to test whether adding segmentation and generative learning elements from the Klingenberg studies, like the existing citric acid simulation, whether it improves outcomes even further. And more generally, closer collaboration between instructional designers and biology faculty during the design process would help ground these decisions in real students' misconceptions rather than design theory alone. Thank you.